Hi, my name is Charley Lineweaver. I'm an associate professor here at the Australian National University. I have two jobs. One is I'm at the Research School of Astronomy and Astrophysics, and on campus I'm at the Research School of Earth Sciences. And for a while I've been very interested in astrobiology and the origin of life and trying to answer the question, are we alone? And today we have a guest who's he's the best chemist I know and who has thought about these questions, and we'll be asking him questions about the origin of life and the chemistry of the origin of life. And this is Steve Benner. Now he is from Florida, Gainesville. He's at the Foundation for the Applied Molecular Evolution.
That's right.
Near Gainesville. And let's say we've overlapped several times at several astrobiology meetings, and each time I've learned something incredible from you. So I thought that we could share some of your knowledge with the audience today. So Steve, why don't you tell us a little bit about yourself as an introduction?
Well, it's good to be here. Thanks for inviting me. That's the first thing. Right, I'm a chemist, so that means that an organic chemist and a biological chemist is sort of how chemistry in my training has progressed. So we worry about how life is put together, the structures of DNA, how DNA might be different if you went to an alien planet and encountered life there.
So DNA being different, okay. I wanted to start out with this thing here. I just got back from a trip overseas, and then in this Delta they give us these peanuts, mini pretzels. And often I say, hey, look at this. What does it say? Enriched wheat flour. It's got thiamine, mononitrate, riboflavin, folic acid. Now all of these things, sodium bicarbonate. And I don't really know what all this stuff is. And I would be the last person to be able to evaluate whether it's good or bad or tastes good. Now, do you know what all these things are?
Yes.
And how does that affect you when you get one of these in your hand?
I don't usually read the label.
You don't read the label either?
No.
Why? I thought that would be one of the greatest things about chemistry. You'd know what was going into your body and coming out of your body.
Well, sure, but it's flour. That means your staple is grass seed, in this particular case, wheat grass seed, I'm assuming. They make the flour rise with the yeast and the baking powder, so they have sodium bicarbonate in there as well. The niacin, these are vitamins, right?
But what, this is there to give us information about whether we should or should not eat it. And you're telling me all that information is useless or something?
Well, it's an interesting question why that information is there. The information is there is because some federal government agency, American or Australian, has required it to be put there. Right. But right, you can, I suppose, go through these things. I would recommend, for example, that you look at the salt.
Oh, okay. There's no salt on here anyway.
Right, the salt. Okay. If there's salt in there, you should sort of know how much salt you're taking in, because as an occupant of the developed world, you eat too much salt.
So as a chemist and your knowledge of the biochemistry of the body, you just look for salt and sugar like you read in a normal health book. There's nothing. Yeah. I would have thought you had some deeper insights into the details that would, I don't know.
If you don't get enough of niacin, you die. Right, you get pellagra. You get these diseases. So in your daily, however, I assure you that if in your daily diet you're relying on Delta pretzels to get those vitamins, you're in pretty sad shape.
Okay, let's forget about that then for a while. Let's talk about the origin of life.
Sure.
Now, we think life on Earth originated, 4 billion years ago or 3.5 billion years ago, and you have been one of the people involved in trying to synthesize life, right? So you take a bunch of chemicals and say, life is made out of this, let's take those chemicals, put them together, and then see what they do. Now, presumably you have not made life in the laboratory.
Well, of course, you're now semantics. What do you mean by life?
Well, what have you been able to make in the laboratory?
Certainly we can make, DNA. We can make DNA that is...
So double helix DNA, does it spiral around, has a helix? How long can you make it? A billion base pairs?
Oh, well, if you put together pieces, you can make a whole genome. And this is what they did at the J. Craig Venter Institute about 10 years ago now. That is, they assembled from pieces all the DNA molecules that are found in a bacterium, a small bacterium, but it was a total synthesis of a genome. Our game, of course, is a little bit different. So we're trying to understand what life might look like if it were not constrained by the history that you and I have had on Earth in 4 billion years past. So there's a constraint coming from origin. Presumably there are only certain ways that you could have actually put together atoms to give you Darwinian systems when life originated, but maybe it will be different on different planets. There are different environments, so maybe it'll be different.
As a specific example, sometimes I get asked by students and others, if there is life elsewhere, will it have DNA or RNA? And you're a chemist, so let's ask you the question: will there be DNA if there is life? Let's suppose that there is life else there. Will it be based on DNA or RNA?
Oh, and it's an excellent question. So we can make in the laboratory alternative forms of DNA and see whether they can have kids. Can they be reproduced? Do they have children with variation?
But that's not what I asked you. You said one is the laboratory. I asked you about outer space. This is the Bob Shapiro question. I don't care what you do in the laboratory. I want to know whether those conditions, are you constraining your laboratory to be like the conditions in the subsurface of Mars or on other planets?
Sure, we try. So we have DNA, for example. You know, your DNA, my DNA, has 4 different nucleotide letters, G, A, C, and T. We've got a DNA that has got 6 different nucleotide letters, G, A, C, T, Z, and P. And we can set that system up so that it has children. So it can make copies of itself. It can adapt. It can evolve. And we can actually get out of that funny kind of DNA all the behavior that you value in modern biology on Earth, but with a different underlying chemistry. Now, we then have to ask the question, is that a reasonable chemistry to have emerged on Mars? What we can tell you is that that's a separate question. Your first question is whether your image of what life could be, is it consistent with chemistry, period, full stop, universal statement. And then you can go back and ask whether the chemistry that you put together could have arisen spontaneously without the hand of God, or for that matter, a chemist.
So, if I understand you correctly, you look at biochemistry, you then say, hey, could it be different in this way, and then you synthesize the differences.
That's right.
And then only then do you start asking the question, well, could this be relevant for life elsewhere?
Right. Now, chemistry's a huge field, right? I can make thousands of different structures. I mean, if I just start drawing the 20 amino acids down and I say I want a 21st or a 22nd or 23rd, it's very easy to come up with additional amino acid structures. So you constrain your thinking, right? So sometimes you'll say, I think I would like to live in a planet with more nitrogen, or maybe with less sulfur, or maybe a planet with more silicon.
Yes.
And so now I can say, I'm not just going to make a 21st or a 22nd or a 23rd amino acid randomly. I'm going to say, suppose I was on Mars. Say, what kind of amino acids might I be using on Mars? And that's the kind of thing that guides what we call synthetic biology at this point, where you're trying to make alternative ways of doing the same things that life does.
But I'm a little bit confused, because I know that the Murchison meteorite fell in 1969, I think they've identified 80 amino acids in it. Only, I guess, 8 of them or so are biological amino acids. And so you have, is it legitimate to say, well, life could have been made out of any of those 80, and let's try to make life out of 20 that aren't even part of the biological ones?
Right, absolutely.
Have you done that? Or?
So, absolutely. So one of the questions, niacin, as long as we are discussing this, niacin is a cofactor. You eat it because, it's a cofactor that does oxidation reduction reactions. And you eat it and you use it in your enzymes because you do not actually have a amino acid that does oxidation reduction reactions like niacin does. So these are cofactors. Same with biotin, which you can also read on the label. This is a cofactor that begins. I don't believe it's on there.
No, it's not on the
Yeah, no, I don't think they enrich that with biotin. But you can get it in your Cheerios, or if you look at your vitamin pill, there will be there. So these are structures that your 20 amino acids have not managed to put into their repertoire. You cannot make a protein with the chemical reactivity that niacin has, and so what you have to do is eat a vitamin and then stick that vitamin binding into the protein. If I were on Mars, I would put an amino acid which has a niacin already in it and make it into the proteins naturally.
But you're talking about something very specific to a human condition, and we're trying to talk about something that's generic to life. Now, famously, Carl Sagan took the ingredients of life, dumped it into a big vat, stirred it up, and say, no life. Now, that's a very crude version of what I imagine you're doing in your laboratory. How much more sophisticated are you? And like I asked before, you haven't created life, nothing's walked out of your laboratory by itself.
Yeah, I mean, life being a self-sustaining chemical system capable of Darwinian evolution. We have done everything in our laboratory except the self-sustaining part. So we have an alternative chemical system capable of Darwinian evolution with 6-letter alphabet, and I'll be talking about that actually over in the School of Chemistry this afternoon. So you can, but yeah, so this is synthetic biology. You set this very grand challenge. Get a set of chemicals that will do something interesting, something that Terran life, natural life does, something that you value, like having kids, evolving, adapting to its environment. But there's no better way to understand this intimate relationship between molecular structure and biology than to try to go out and make a biology of your own with your own molecules. Okay. Now, self-sustaining is hard. We don't want necessarily to have this stuff be self-sustaining, because then it can escape from the laboratory and do nasty things. But we certainly have done everything short of that at this point.
How about taking the approach of looking at like the phylogenetic tree of life on Earth, and then seeing what the earliest ancestors were, I don't know, 3.5 billion years ago or so, and then saying, okay, that's simpler than all the complicated stuff we have around us today, so let's try to make the simplest life we can. Is that a strategy that you guys use?
Well, other people do it better than we do it. So, sure, absolutely. You have an example of life on Earth. It's one example. It's all descendant, as far as we know, from a common ancestor. But sure, you can go try to infer from the diversity of life on Earth what the ancestors were and how much simpler they were. And if you do that, you can come back to a sort of an idea of what the essence of life is. Of course, if you want to be lucky, go to Mars, you can actually discover a second example of life, and that would be an enormously revealing discovery. You can go back and try to simulate the origin of life to try to understand constraints on origin that chemistry implies. But then you do what we do, which is try to make alternative, try to get the second example of life in the laboratory rather than hoping to get lucky by finding a second example on Mars.
So if we do find a second example on Mars, I mean, are you going to go take at it with your chemistry set and say, yes, it is life, or no, it's not, or...
Well, let's assume it is life. We found a second example. That poor life form is going to be dissected. It's going to be dissected down to its individual molecules. We would try to find out how it works. So this is your friend and mine, Paul Davies, who was looking for a second example of life hidden in the shadow biosphere on Earth. These guys really should be hiding from us because we will be interested in knowing how that works, and almost certainly will begin with our dissecting. That's a bad thing. But
occasionally I run across the question of chlorophyll.
Yes.
And we say, hey, the spectrum of life coming from the sun has a peak at around green, and yet green is what is reflected from the chlorophyll. And so the question is chlorophyll just such a hard molecule to make that only there can it do what it needs to do to be efficient at converting sun photons into chemical energy? Or is it, could it have been something else? I mean, should we expect chlorophyll on planets around Alpha Centauri, for example?
Right, no, but that's right, exactly. So, I mean, this is the book that I wrote on life, the universe, and the scientific method, was asking the question, how would you go about, as a scientist, trying to guess whether there are extraterrestrial life forms and what they might look like? But yeah, you're absolutely
right. But chemistry
right.
is not infinitely flexible.
It's not infinitely variable.
But you know where it is and where it's not flexible. And that's the kind of thing that I wish I knew. And so I want to come
something about it.
Well, I want to come to what I call Benner's problems. So Benner's problems are why. You said water is a problem.
Yes.
Okay, you have a whole list of things about why it's hard to make life, and I think ribose is a problem.
Yeah,
so
sure.
could you tell us this list of problems that you often tell us about?
Right. Well, before we get off of chlorophyll, you're absolutely right. You can look at any of these features of modern life that we know. I mean, physiology, why do you have 5 fingers, not 4 like the Simpsons, or 3 like a dinosaur?
But some of them will be quirks, and others will be very fundamental.
So some of them reflect current adaptation. They are best for the world that you live in. Some of them are vestigial, right? They reflected some past adaptation. Some of them are just historical accidents, right? And you can ask that same question about the structure of chlorophyll. And, if you say, I'm going to look at the power law, the irradiation of energy from the sun, is chlorophyll perfectly matched from that? You can make an argument one way or the other, and people do. And if it's not perfectly matched, you have to say, it's not optimal. That means there is something constraining the structure from being optimal, and chemistry can be one of them, right? Chemistry can
So
be
what's the
a
answer?
problem. Well, so for example, water is a
No, what's the answer with chlorophyll?
Well, that's a good question. I mean,
Give us your thoughts. Give us a brain dump on
I
chlorophyll.
would, yeah. I mean, I would have to get you a structure of the molecule and draw it out. So it's an elegant structure, right? So it has magnesium in the middle, right? That's an important part of its function, and that's.
But magnesium is all over the universe.
Exactly. It's
Okay.
not a bad choice because it's relatively abundant. If I had to put gallium or some exotic metal in the middle, I wouldn't have enough of it to do it.
But do you think that's but we have bacterial rhodopsin, which is also gets energy from photons,
That's right. But now iron, right? These are common elements. But think about iron for a minute. Iron is very easily accessible on an Earth which does not have an atmosphere of oxygen. The minute you have an atmosphere of oxygen, iron gets oxidized, it becomes rusty, and it precipitates, it becomes inaccessible. So you can go back and look at, for example, the use of iron all over the place in your metabolism, and you might even find a supplement in your dietary material. And that iron is there, right, instituted in metabolism back 2.5 billion years ago before there was oxygen.
Yeah, but how do you ask the question, iron was there, but was it coincidental or was it obligatory? I mean, that's what we're trying to do. Could I have easily put in something else or not?
Yep.
And that's what you try to evaluate.
That's right. So in the laboratory, you would try to make chlorophyll with aluminum. It fails, right? And so magnesium becomes important. And that's a constraint from chemistry, right? If I try to make it by putting germanium in, that's a constraint from just natural solar abundance of materials, right? I don't have.
But there's
enough
chlorophyll A, chlorophyll B, there's about, 6 or a dozen types of chlorophyll. They all have magnesium in them?
Yeah, absolutely. And so you now can look at the chemical structure of chlorophyll, and you can see in that chemical structure vestiges of what are most easily interpreted as prebiotic chemistry, right? You can assemble in prebiotic kinds of environments, at least what you might think, the ring around the magnesium, which is the chlorophyll. And
can you make a different ring? Use magnesium, different ring?
But not so easily prebiotically, right?
Not so easily prebiotic.
So you can look at that. Well, now, I would be a lot happier saying that if I knew what prebiotic environments were like. At this point, we can only guess. But absolutely, you can go look at these structures of these molecules and you can see features in them that look like they're recently adapted, which means that you've gotten pretty sophisticated metabolism over the last couple billion years, so you can get what you want and you've gotten it. But then you can see some features in those molecules that look very ancient, and some of them even look like they've been constrained.
Yeah.
Like adenine and
but
bases.
new, adaptation, and ancient, that's not really addressing the question of whether it could have been different. I mean, it's true that if you had something ancient start out that way, well, is that obligatory for other planetary life or not? That's the question.
That's right. So when we go back and try to address this kind of question experimentally, the first thing we start by doing is trying to say, there might be a different structure. Then we say, okay, does it work chemically? So adenine is a base, GACT in your DNA. Adenine is the A. It's made prebiotically from hydrogen cyanide. Hydrogen cyanide is abundant in the cosmos.
So adenine is abundant in the cosmos, I suppose.
That was the argument. So you would not be surprised to find adenine or one of its derivatives used in Martian DNA.
Well, let me turn the question around a different way then. I'm an astronomer, so what is very, very abundant in the universe that is not used in life? I know helium is one answer to that question.
I guess that's true. Helium certainly is.
But how about something else? It's falling down with all these amino acids, and it's there. And I think Miller used to say, "Hey, here's an amino acid that's very abundant, but it's not used at all in life." Sure. Alpha, beta. I forget what the name of it was.
That's right. The beta amino acids are very abundant in the meters. Look, I mean. Each of these is a just so story. So without going into a structure, drawing on a blackboard, you have alpha amino acids, and what that means is the amino is right next to the acid. So you could have a beta amino acid, which means that the amino is not right next to the acid, it's a little bit farther
away. We're all alphas.
You're all alphas.
And why is that?
And that's because, according to the story, because if you make a protein out of beta amino acids, it's too flexible and floppy, it doesn't fold very well. Whereas you make it out of alpha amino acids, it folds. And folding is important to get out of your proteins a catalyst or a receptor or a binder, this type of thing. So folding is essential. I mean, the proteins start off as a linear string of amino acids. You've got to fold them into a three-dimensional structure. Alpha amino acids do, beta amino acids do not. And so there's a chemistry constraint which trumps the availability in prebiotic cosmos constraints. There's more beta amino acids around in the Murchison meteorites, shall we say, but you don't And then so if I were just going to use what is abundant and use it in the proportion of its abundance,
Yes.
I would use beta amino acids. But then chemistry steps in and said, yes, that's true about abundance, but the constraints of function require that I use the scarcer nucleotide. So, the chemist in the laboratory is the person who's actually going to try to make a protein out of the beta amino acids, or a non-chiral collection of building blocks is one of the things I know you are interested in. The chemistry tells you what constraints are, merges and mirror, it tells you what's out there, and what you're looking at here's this dance between function and availability, which is somewhere going to lead you to a model for
All right,
elections.
let me ask you one last question.
Sure.
We're starting to run out of time, but this chirality, this homochirality is sometimes considered to be very fundamental and sometimes not. And we have left-handed amino acids and we have right-handed sugars.
Right.
Now, have you tried making left-handed amino acids with left-handed sugars? Or right-handed sugars with right-handed amino acids, or vice versa? There's 4 possibilities there, I guess.
No, I understand. I mean, one of the things that's very hard to explain in a short interview without actual physical models is what's a left-handed and right-handed species is a convention that is defined by a chemist. What we
Right.
can say about the amino acids...
No, my question is, when you have a right-handed sugar, does that mean, is it preferentially like left-handed amino acids, or could it just as easily do with right-handed amino
That's right.
acids?
You really can only make meaningful discussions about left-handedness and homochirality within a class of compounds. So if I start to move from amino acids to sugars, it's purely a convention to what I call left-handed and right-handed.
So
that's right.
So could I then make life based on
Right.
left-handed sugars and left-handed
Right.
amino acids?
And this is one of the things
Can you do that?
Yes.
Have you done that?
Well, no, of course not.
Why not?
No, but what we can tell you is that if I'm going to build a polymer out of amino acids, they all had better have the same chirality,
yes, of
no
course.
matter what I call
Okay.
them, because that's the only way that polymer will be evolvable.
Right,
That's
but
the
polymers
only way.
talk to each other, and that's why I thought that...
But that's right. But look, I mean, it's what we confuse our organic chemistry students on. So glucose, when it is being converted into vitamin C, you oxidize one end and you reverse the other, and it becomes D-glucose, actually becomes L-gulose. And you haven't changed stereochemistry of anything. You just change what numbers you give in terms of priority and deciding. So why do you call that your left hand? It's only because you come from culture, right?
All right, so last question.
Yeah.
Do you think there's life elsewhere in the universe?
I do.
Because...
Well, if you look at Mars, I actually think that there's a very good chance that there's life even today subsurface on Mars. And the reason for that is because there were oceans on Mars 3 billion years ago when there was life on Earth. And, it's an interesting problem. I mean, the idea that you have a singularity on Earth this far ahead of the expiration of the sun would be quite a surprise to me. If life were scarce in the cosmos, we would be having this conversation 15 minutes before the sun, proceeded to extinguish. And so these are these arguments made on the statistics of very small numbers. We have one example, but we know that life is emerging on Earth well before the sun is even close to being extinguished. If life were hard to emerge, you would only be getting life emerging when the sun was near extinction. And so these are the kinds of arguments that make me confident, but they're not. It's not a justified confidence.
Okay.
It's a weak confidence.
Right. Well, you should read my paper on that.
I have.
Anyway, Steve, thank you for coming. Thank you for
pleasure.
answering the questions, and we're looking forward to your lecture. It's about half an hour from now.
That's right.
Thanks for coming.
My pleasure.
All right.